lesson, 15.4 is all about RNA RNA processing. So the RNA polymerase attaches. Which RNA polymerase is it?
2.
Attaches and unzips and places RNA nucleotides onto the DNA chain. Click. Click. Here's a couple. Click. And it just keeps doing this and running down until it has an RNA, what we call an RNA transcript. Oh, and by the way, it always goes in the 5' to the 3' direction. Just like when we copied DNA. Are y'all with me? It seems like you're not, Thomas.
Highlighting on mine.
Oh, okay.
15.4.
Attach in the 5' to 3' direction, then this breaks off, and the DNA will come back together, so we conserve the original DNA strand. And now we have this piece of RNA here. And do you remember the 3 things we did to it? There's 3 things that we do to the RNA transcript. Let me make it a little longer.
Take out the introns.
We remove the introns. This is an enzyme called a spliceosome. Comes in and it grabs the introns and it pulls them out. And then what's left? What do we call the things that are left? The exons come together, and it kind of shows how it works in this picture here. See this? This spliceosome is actually 4 or 5 different proteins. I think it's 4 different proteins. They got a cool name. They're called SNRNPs or SNRPs.
That's nice.
And the little SNRPs come together and make one enzyme called a spliceosome, and that pulls the ends. This green part here is all an intron. We got to remove that. So the spliceosome attaches and pulls it together, and it makes kind of a loop. And this part attaches to that part, and you see it kind of loops around and attaches, and it makes this exon and that exon come together, as you see here, and this intron is left, and it just kind of floats away, and later gets torn up by the cell, and the nucleotides get reused. So that is the removal of introns. You want to see video footage?
Thank you.
Very much.
Video footage of intron removal.
Transcribed into RNA, it needs to be edited to
Hold on. Start over.
As DNA is transcribed into RNA, it needs to be edited to remove non-coding regions or introns shown in green. This editing process is called splicing, which involves removing the introns, leaving only the yellow protein coding regions called exons. RNA splicing begins with assembly of helper proteins at the intron-exon borders. These splicing factors act as beacons to guide small nuclear riboproteins to form a splicing machine called the spliceosome. The animation is showing this happening in real time. The spliceosome then brings the exons on either side of the intron very close together, ready to be cut. One end of the intron is cut and folded back on itself to join and form a loop. The spliceosome then cuts the RNA to release the loop and join the 2 exons together. The edited RNA and intron are released and the spliceosome disassembles. This process is repeated for every intron in the RNA. Numerous spliceosomes, shown here in purple, assemble along the RNA. Each spliceosome removes one intron, releasing the loop before disassembling. In this example, 3 introns are removed from the RNA to leave the complete instructions for a protein.
Pretty cool, huh?
Yeah.
Yeah.
It's happening in your cells right now.
Wait. Oh, wait.
Every cell, in every cell. And you have 80 trillion cells, by the way.
What if the cell's in it once it's G0? It's just
No, it's still doing that because your cells,
It's gotta stay
G0
alive.
still have to make protein.
What hap-
proteins.
What happens to
If
the
the cell's
edited part?
alive, it's making proteins.
Right.
So
Think about that.
every living cell. Now your dead cells aren't doing it. Yeah.
What happens to the edited part that isn't?
That floats away. There are enzymes, other proteins that come in and tear it apart,
Okay.
so that the cell can reuse the nucleotides. It still wants the nucleotides. That's useful. Now the other thing, so now that we've removed the introns, what do we do with the rest of it? There's two things.
You need a guanine cap.
Guanine cap. There's modified guanines that are added to the front, to the 3' end. That's called the guanine cap. I'm sorry, to the 5' end. And what else? There's other enzymes that add what's called a polyadenine tail, and these enzymes will put on an adenine and another adenine and another adenine, and that goes on the 3' end, and it makes this long adenine tail. And it might be 50 adenines long. And scientists hypothesize that the longer the tail, the more proteins this tRNA can make. The shorter the tail, the less proteins this tRNA can make.
I have to go.
Bye.
Bye, Josie. Bye.
She's very demanding today. Made me turn off the car, and then it's just demanding.
I always got your back for a bar. You know that.
I'm kidding.
I know.
Thank you. All right. So now we have this long strand of tRNA that we're going to move out of the nucleus to the ribosome, and that's the next section. And I've made a special one on the string. It's easier to move. Turn the light back on for this. Always wanted a remote control light.
That'd be so cool. What are the ones that you clap and it turns on?
Clap on, clap off, clap on, clap off. The clapper.
That'd be so cool.
Y'all remember that commercial?
I do.
Big in the 70s. All right. So here comes the tRNA out of the nucleus, and it's got its little cap, and it makes its way out here to the ribosome. Now the ribosome has two subunits, and like I said, the ribosome itself is made of RNA. We call the ribosomal RNA rRNA. The reason why I'm getting ahead is so y'all can do this with the magnets on the board tomorrow. We're going to set them up on all the boards. Okay? But pay attention because you're going to have to show this to me. So the ribosomal RNA is kind of coiled up RNA. There's also proteins in it. It's a big structure made of RNA and proteins. And it's got two subunits. It's got a small subunit and a large subunit. The small subunit attaches first, and the large subunit, and actually they're separated, but they'll float around and this thing attaches later. And now you have the intact, full ribosome. And these things are also important. These things I've got right here. Let me move this down a little bit. I got it up so high. So this is rRNA. What's this piece called right here? It has a name too. mRNA. That stands for messenger RNA. It's bringing a message from the nucleus to the ribosome. It's a copy of the DNA. What's the only difference between this copy and this half of the DNA? U's instead of T's. This goes ATG. This goes AUG. Huh? Exciting?
Super exciting.
And now these things, you got to know what these things are right here. They're called tRNAs. These tRNA molecules, tRNA stands for transfer RNA. And this is what it actually looks like. It's a little bit different on my model. But it has a part down here at the bottom that we call the anticodon. And the anticodon, I have real big here. It's actually not bigger than the rest of the nucleotides, but it needs to be big for this model to work. But anyway, it has 3 letters on it, and that anticodon will attempt to attach to the codon, which is the first 3 letters of the mRNA. So here's the codon. This is called a codon, and this is called the anticodon. The anticodon of the tRNA attempts to match up with the codon of the mRNA. Now they call it tRNA because it transfers an amino acid. Each tRNA carries an amino acid. It's probably important for you to know how the tRNA gets the amino acid. There's an enzyme that has a funny name. It's called Aminoacyl tRNA synthetase. And why I say it's funny is I had a professor at Georgia Tech that was teaching us this, and he wasn't from America. He was like from India or somewhere, and he couldn't speak English very well. And he would say, it's called aminoacyl tRNA synthetase. He would say aminoacyl tRNA synthetase. Aminoacyl, he would say that. And he said it during this one lecture like 30 or 40 times. And every time he said it, everybody would laugh. And we had this huge lecture hall with like 200 of us. And he kept saying amino, and he didn't know why everyone was laughing, but he got happy and everyone was happy, and he thought they were just happy to be there, I guess. So anyway, I'll never forget aminoacyl tRNA synthetase. And what it does is it takes an amino acid and its active site right there, and it hangs onto it, and the tRNA will come up and will attach and will pick up that amino acid and take it away. You see how that works? You want to see again? You're going to have to show me this tomorrow. tRNA comes up, attaches to aminoacyl tRNA synthetase, and it comes away. And now the correct amino acid is attached to the tRNA, which you can see it up there at the top on this thing. You see right up there at the top. That's the amino acid that's carried attached to the tRNA. It's stuck there at one end. And now, here's the thing you have to wrap your mind around. Each tRNA can only carry a specific amino acid. In other words, this amino acid is proline, and it only fits on this tRNA. I've called it tRNA3, but those numbers are just for the magnets. It's not really called tRNA3. But it's a tRNA that will just carry proline. So there's a specific amino acid that fits to only this tRNA that has the anticodon GGC. The anticodon GGC codes for proline. You see what I'm saying? There's actually a chart. I wonder if I have it here. I have my chart. So, I want you to look. GGC. Now, GGC is the anticodon. What would the codon it attaches to be? What goes with GGC? CCG is the codon it would attach to. And what codon does CCG, what amino acid does it have? Proline. See? This GGC anticodon has proline as the amino acid that tRNA carries. So if you run into a CCG sequence, you're actually coding for the amino acid proline. every codon codes for a particular amino acid in this way. And by the way, there's a different aminoacyl tRNA synthetase for every tRNA. So if there's 64 three-letter combinations, then there's 64 different tRNAs carrying the different amino acids. And actually that's not quite true. In other words, for instance, this one that codes for proline, CCU codes for proline, CCC codes for proline, CCA codes for proline, CCG codes for proline. It really doesn't matter what the last one is. I think it is correct that the same tRNA it might be that this last nitrogen base here is just not even there. And you only have to have the first two attached. And the last one doesn't even matter, in other words. So you might only have one tRNA that carries this proline instead of 4 different tRNAs. But anyway, tRNAs carry amino acids, and they have specific anticodons. So let's see how this works. AUG is the first codon here. Did y'all know AUG is the start codon?
Yeah.
What does it code for according to the chart? Can you find AUG up there? Met. That's methionine. That's the first one. And so what will happen is tRNAs are bouncing around in the cell. They're bouncing around, and they'll come in here, and they'll come in here, but that one won't attach, so it just bounces out of there. And this happens very quickly in nanoseconds, billionths of a second. It can bounce in here and attach and come out. And another one can bounce in here and attach and come out. And another one can bounce in here and attach and try to attach and come out. But this one bounces in here and it has the right sequence. So it does attach and it stays there. And the first amino acid, which is methionine, is in place. Chaz, you getting all this? Now, the second amino acid, the second codon is here. And it happens to be ACG. So what would attach to ACG? If you're RNA, what attaches to A? U. This is a C, so what attaches to C? G. This is a G, so what attaches to G?
C.
So it would be UGC. So something that has UGC comes along.
On the bottom.
And let's say this one comes in and tries to attach, and it doesn't.
Over here.
Where is it? Oh, thank you.
Yeah.
And here this one comes in and tries to attach, and click, it does. And now you have 2 tRNAs side by side inside the ribosome. And then what happens is an ATP, an energy molecule, comes in and attaches to the ribosome in such a way that it gives some energy and causes a bond to be made between the 2 amino acids. You know what we call a bond between 2 amino acids?
Peptide.
It's called a peptide bond. Peptide bond. So we bond the 2 amino acids together.
How big did you say the tRNA was in relation to ribosome?
They're similar in size. I think the tRNA is smaller than the ribosome by a little. I'm going to show you a video that shows them in the correct size.
Is it in the ribosome when it's doing this?
Yes. Yeah. The ribosome is actually a 3-dimensional structure, so it actually comes out at you. And so this form, this large subunit and the small subunit form like a room, a chamber. And these tRNAs will come inside of it. So the tRNA is smaller than the total ribosome. It's small enough to fit inside it.
Maybe not the subunits.
Maybe not the subunits. That's a good question. I don't know the answer. I'll show it to you in a video here, maybe we can figure that out. Okay, so what happens next? The ribosome slides along like this and moves one codon down. Josh, you getting all this? You seem as if you're asleep over there. We move one codon down, and when we do that, this first tRNA will exit. And there's actually sites in the ribosome. I don't know if you can see this. The E site, the P site, and the A site. The E site is where the tRNA exits the ribosome. And it'll exit the ribosome, and it'll bounce around in the cytoplasm of the cell until it runs into an aminoacyl tRNA synthetase that has the correct shape so it can fit and pick up another methionine. And so it can go again. Once it picks up another methionine, it's ready to go again. So these things are just kind of couriers. They're like taxi cabs that bring the amino acids to the right spot. Questions yet? Now, there's two other sites in the ribosome. There's the P site and the A site. We call this the P site because it's where this protein or polypeptide is residing. So this is a forming protein. We're building one of these chains, right? Y'all with me? This is way too exciting for y'all not to be like.
Biden was just sworn in.
What's that?
Joe Biden was just sworn in.
Oh, okay. A little bit off the subject. I guess the cells are making proteins, though.
Yeah.
So,
Where are they?
all right. So here are the here's the protein forming. Listen, the P site has the protein forming, and the A site. What's the A site? The A site is where the new amino acid comes in to the A site. That's at this side, and another amino acid will come in and bind and form a peptide bond. And then the ribosome moves down, and then the tRNA exits from the E site. And does it just go and get any amino acid?
No.
It can only get the one it dropped off before, which was this red one, threonine. And it'll get it from a specific aminoacyl tRNA synthetase. And now we're almost done here. We got another, you see the growing polypeptide here. We have another codon exposed, it's CAU. It'll fit with this tRNA. I know because I have them numbered. Click, bond, slide, leave, get. Now we have this final. We did that. I only have 4 amino acids here, but a normal protein might have 400 amino acids.
Whoa.
And this is going like 10 amino acids per second. And so we get to the final thing, and what's this final thing here? This is UGA. Can you read what UGA is up there?
Said stop.
It's a stop codon. UGA is one of the stop codons. I never forgot that because UGA, University of Georgia. Stop. I don't want to hear it. I went to Georgia Tech. Stop. So UGA.
Bradford's here?
No, she's older than me. UGA connects to a release factor, and that release factor causes the ribosome to slide over, this tRNA to leave. This tRNA leaves. It goes and gets another purple one, and then the ribosome actually breaks off, and these parts float away, and they can come back together somewhere else later and make another protein. and the release factor leaves, and there's your protein. It's just sitting there in the cytoplasm, and it will fold into its final shape. And Andrew's not listening. And then the tRNA still remains, and it can go through another ribosome over and over and over. And that's the whole translation
Wait,
process.
can any ribosome do, or is there like specific ribosomes for proteins that we have to do?
Any ribosome in the cytoplasm can transcribe this tRNA, or this mRNA. Not transcribe it, can translate it.
Okay.
This whole thing called translation.
How does it know whenever it's done with the mRNA?
They're not sure about that, or at least I don't know if they've discovered in the last 10 years or so, but since I'm in the last decade or so, they think that every time it goes through, maybe one of the A's gets knocked off the end, and after you're out of A's, it won't work anymore, and the whole mRNA can get destroyed by enzymes. Hold on, don't pack up. I got a video footage of it. Here's what it really looks like. This is a great video.
I really hope I'm not disappointed. I hope Andrew's in there. I hope you're in there. This is your stuff. This is your stuff.
Protein synthesis, translation.
That's not the one either.
That's not.
Like got a weird title. Like high school.
This is that.
Second biology class or something.
This is what Francis Crick
I don't know, it's big
called
money.
the central dogma of modern biology. How DNA makes protein. It starts with a bundle of factors assembling at the start of a gene.
So this is
It
transcription.
leaves that trigger the first phase of the process, reading off the information that will be needed to make the protein. The gene is the length of DNA stretching away to the left. Everything's ready to roll. 3, 2, 1.
Okay, go.
Yeah,
The
this
blue
is
molecule
transcription.
racing along the DNA is reading the gene.
It goes into translation.
It's unzipping the double helix and copying one of the two strands. The yellow chain snaking out of the top is a copy of the genetic message, and it's made of a close chemical cousin of DNA called RNA. The building blocks to make the RNA enter through an intake hole. They are matched to the DNA, letter by letter, to make an exact copy of the A's, C's, G's, and T's of the gene.
Watch the video, David.
The only difference is that in the RNA copy, the letter T is replaced with a closely related nucleic acid known as U. You are watching this process, called transcription, in real time. It's happening right now in almost every cell in your body. When the RNA copy is complete, it snakes away from the nucleus and into the outer part of the cell. Then, in a dazzling display of choreography, all the components of another molecular machine lock together around the RNA to form a miniature factory called a ribosome. It translates the genetic information in the RNA into a string of amino acids that will become a protein. Special transfer molecules, the green triangles, bring each amino acid to the ribosome. The amino acids are the small red tips attached to the transfer molecules. There are different transfer molecules for each of the 20 amino acids. They all carry a specific 3-letter code that will be read by the machine. Now we come to the heart of the process. Inside the ribosome, the RNA is pulled through like a tape. The code for each amino acid is read off, 3 letters at a time, and matched to 3 corresponding letters on the transfer molecule. When the right transfer molecule plugs in, the amino acid it carries is added to the growing protein chain. Again, you are watching this in real time. And after a few seconds, the assembled protein starts to emerge from the ribosome. Ribosomes can make any kind of protein. It just depends what genetic message you feed in on the RNA. In this case, the end product is hemoglobin. The cells in our bone marrow churn out 100 trillion molecules of it per second.